Thanks very much. I'm just going to turn on my little timer. Yeah, hello, I'm Sebastian. I am from Montreal, from the Origami Lab at the MNI, or as we now call it, the Neuro, which probably makes a lot of sense. I am filling in for Michelle Wang, who's also from our lab and can't give this talk. I am clicking. Because we all use Stefano's template, I also have a conflict of interest to declare, which is I am a member of the Neurobegal and NIPOPy communities, and I like these tools. And later you will hear about them, but not only about them. Okay, so I'm talking about reusing and sharing and doing this sustainably. So where's the link? I mean, Nikhil already gave the motivation in a way. If you don't have to do processing a second time because you can use it from somebody else, that would be great. So there are a couple of words that we use for, I guess, general recycling. So we say, don't produce as much, which would don't run it again. Reuse somebody else's things. And repurposing, I guess, would be things like Open Neuro, right? Somebody collected a data set for a specific purpose, makes it open, but you're using it for a different purpose. That's a good thing. There are other reasons why we would do this. Excuse me. So one is expensive. If you're using cloud, you pay for this, or your university does, or your grant. So that's one reason. Also, time and capacity. So either you may not have the time to run processing. If you're looking at UK Biobank and you want to do an analysis and you're starting with the raw data, you're not going to have a lot of results if this is like 3 days before the conference. And capacity means sometimes you're not the expert on it, right? So maybe you're a machine learning person, or you're doing primary statistics, and what you're interested in are tables. You don't want to look at brains, and you don't understand how you get from brains to tables. So that's the capacity side. And then the thing where my conflict of interest, if you want to call it that, or our interest comes in is really ethics and governance. So what if you can't have the data? How do you process somebody's data if you can't touch them, right? Well, that's not so easy. So what you have to do in order to do sharing and reusing is you have to find other people, and then you have to work with them. And usually when that happens, you need some type of coordination. And that's what I'm trying to talk a little bit about. So you probably heard of FAIR. I'm from Canada, so I'm calling this part the fir tree, because I'm not talking about access. So we have these in Canada. And basically, FAIR means findable, which is the last thing I'm going to talk about. That means how can people find my data once I've processed them and then they want to do things with them. Interoperable means how can I combine data sets? How can I make tools that can work on different data sets? And we've already heard some of the solutions for this. And then reusable is a way of, if somebody's made a thing, what does it take for me to be able to benefit from that, to reuse it in my analysis? Like, what are the things that they would have to do or you do for somebody else so they can reuse these tools? Okay, so if we're looking at the process, we've seen a similar process figure. At a very high level, you collect data, and then at some point you do analysis, and the way I'm going to talk about it is you curate, you process, you find, right? So what are the challenges here? Well, first you have to collect data. I'm not going to talk about that, but we can imagine that usually in imaging we have tabular data and we have imaging data, and then they're being collected somewhere. And then that's where typically our life starts. I don't know, maybe you're also collecting data. Then the first question you could ask is, well, suppose you have friends and you want to collaborate with them, and now either they give you data, or you want them to run your processing pipeline, or you want to do some kind of coordination here, right? What is a way for you to do this so you can more easily do the processing? So if everybody sends you a data set, how can you make your own life easier if you want to process all these data sets? And it turns out that's a hard problem, and it's not a new problem. So I'm trying to group it in essentially 2 problems. So how do you understand, or another human understand, the data that you've just been sent, just been given? And then the other question is, if you have a tool that's supposed to do something with the data, how does the tool understand the data? And you'll see how slightly different, let's say, levels of organization are needed. So if this is a DICOM data set, how do I make it more standardized? So I'm briefly going to talk about one of the earlier efforts for data sharing, which you're probably familiar with, INDI. So really the spearhead, or whatever you want to call that. So the way this worked was they got a bunch of people together and said, can you please all pull your data, and then we'll process them, and then that's how we get 1,000 functional connectomes. Now, the tricky thing is, they said, well, ideally you organize the data in this way, but really, if you don't, that's also okay, we're gonna clean that. And if you've ever been in a position where you have to clean data that is sent to you from other people, you are not gonna enjoy that process because it's quite tricky and you have to understand these tools, but it's doable, right? It's doable. And I guess my analogy, I've been traveling quite a bit to come here, and I've visited many friends, and so when I try to help them set the dinner table, right, I have to ask them, because every kitchen is different, like where are your forks and where are your spoons, and then they explain it to me, or if they're not home, they write a note, and that works. But that's kind of the human side. If you imagine I had this really interesting blender that I wanted to bring to all my friends and make them smoothies. The question I would ask is, does my blender work in your kitchen, right? So if I'm a tool developer, that's really when I'm getting to the point of standards, right? So I want to know, does your electrical outlet, or maybe not the blender, maybe it's your laptop, does your electrical outlet fit my tool, right? So if I'm doing things by hand, I can write it in a text document, and that's okay. I'm sure this, I promise you this is going to come back to neuroscience. But if I'm developing tools, that's really when I want standards. And so we're bringing this back to our field. This is when you have the standards for apps that can consume these data systems. We've already heard about BITS. So BITS is the brain imaging data structure. I'm just going to give you a very brief overview. I'm assuming most of you are familiar with this. But really, BITS, the idea of BITS is it's good for humans and for machines, right? So both you understand the data and the tools do too. So BITS is a standard that tells you how to organize and name your data, your files in a data set. So it has a directory tree structure, and it has a naming convention for the different files. So that means if you get a new BITS data set, it should look familiar to you in terms of the data organization and naming. It also encodes metadata in this key value pair structure. So if you look at a BITS file, you will notice that there are these recurring prefixes, so to say, right? So the way it makes it a key value is that sub is the subject, and then the value part behind the dash would be the number of the subject, or the ID of the subject. And then session, same thing, is a key, and then the value would be 1, and then this is a suffix that tells me it's a T1 weighted scan, for example. So that's how BITS puts order into the chaos. Another thing that BITS does is it tells you what file formats to use, right? So if you're imagining tabular data, there could be many, many different ways you save a table. You could use a TSV, a CSV, an Excel file, or a PDF, right? And so BITS says, how about we just all use this one format, and then we have an easier time working together. And it does that for different types of data. So imaging files are nifty and so on. And then the last part is there's a lot of metadata around data. So what does it take to make the imaging file that you're sharing with somebody interesting is usually that you can describe what this file is about. And so BITS tells you where to put that type of information. So there's this idea of a sidecar file, which basically is a text file, a JSON document that contains additional information about a binary file. So for example, you have an image file, and then you have this JSON sidecar file that tells you, oh, this was a 3 Tesla acquisition, and this was the flip angle, and all these things. Or maybe it's a table, and it tells you what's in the table. Okay. Again, I'm assuming you are mostly somewhat familiar with this. Otherwise, please ask questions. Now, the last part is you have to somehow go from this part to this part. You have to go from your input data to BITS, and lucky for us, there's a very vibrant community of tool developers. We've already heard about EasyBits. So there are other tools that help you convert from DICOM to BITS, and there's essentially these two parts, which is the file format conversion from DICOM to NIfTY, and then also the file data organization conversion. Okay, so essentially the answer to the question, how do I curate my imaging data, is turn it into BITS. That's the best standard that we have for making processing easier. And so now, if all your friends have organized the data into BITS and you're going to the processing stage, then you're saying, well, okay, how do I now process the data in such a way that we can more easily combine and integrate it and reuse it? And so lucky for you, because your data are in BITS now, you can make use of the entire ecosystem of BITS apps. So these are all pieces of software that developers have developed that consume BITS datasets. So that's what I was talking about. This is one of the big benefits of standards. But there are essentially two problems with this kind of reusability, right? So one question you're asking is, if somebody gives me a data set, what did they do to create the processing output? And is what they have done compatible with what I'm interested in, right? what kind of confound regressions they did or what kind of version of pipeline they ran and so on, right? That you want to know that information because that's probably how you base your decision on including it. And then the other side of it is if I run processing and then I want my friends in another institute to do the same thing, what do I have to tell them so they can do that? And again, you can think of different ways. I'll show you a couple of doing this. So you first look at what did they do. So the tricky thing is, even if I tell you I've run this specific tool and I picked this specific version, and you've heard about containers, so they usually take care of that part, these tools have options, right? So you can run them in different ways. There's like a flag, and that does a slightly different thing. So parameters is another way of calling them, and we need to know which parameters we use to run a thing. Or provenance is another way of saying this, essentially a history of what was done to the files. And the tricky thing with history is that we're not very good at writing this down. I mean, maybe you have done it, but if you're looking at a method section, right, there's all kinds of degrees of, let's say, exhaustiveness of how people describe the exact parameters and steps that were done. And maybe there's a script included, but the problem with that is then everybody writes it slightly differently. And if you run it again, you maybe forget to update things. So we're not the best person to do this kind of provenance tracking is, I guess, what I'm trying to say. And so here's another example. I think this is also from the FCOM 1000 people who, wrote down how these data sets were processed, right? And I mean, it's very useful to understand what they were doing, but it's not a complete exhaustive list. You would have to look somewhere else for that. The other thing that I just want to point out, even if somebody writes down an exact record of what they did, it doesn't necessarily mean you can reuse, like re-execute it, right? If I have a big text file that says, here are the following steps that were done to make this file, and then I try and do that exact same thing again, I'm probably going to compile human written text into something that's executable by a computer, right? So the point I'm trying to make here is, again, there are tools, and tools are good if they make such tasks easier for us. So you're familiar with Git because Stefano told you a lot about the benefits of Git. And so Git is a way of controlling the versions or the history of code. So if you change a text file, you can say, here, I made a change, and then the exact change is logged, and then you can write a little comment on why you made that change. And you can imagine that doing the same thing for data or data sets would be very useful. And indeed, there is such a tool. So there's a underlying thing that's called Git Annex, but Datalat is a tool that essentially lets you record a history of everything that you did to change or create a specific file. And the way it does this is by using Git. So essentially the history that you're seeing here on the right, that's a Git history. So the command here is actually git log. But what you're seeing inside the history is not just I created a list of podcast titles, which that is interesting, but specifically which script was run, and it's a very exhaustive provenance record in the sense that you not only know the command that was run, but also which data were the input, and then which file was the output, and then if you run it again, it'll keep track of that as well. So it's a very powerful system. And the way you use it is you install it. And then every time you want to run a thing, you say datalat run, and then that's how datalat knows you just made a new, you ran a new command essentially, and it writes it into the history. So again, very powerful. I think it would be fair to say that it's also technically complex, and it's something that is very domain general. So it's not specific to neuroimaging. But if you have a system that can use Datalat for you to keep the provenance record, that's very useful for you to know exactly how a file was created. So in the neuroimaging community, there are other projects that use Datalat for different things. So we've heard about Datalat as a way of moving data. That's another thing you can do. I would just want to briefly tell you about the BIDS apps bootstraps or BAPs. So this is essentially a framework that kind of takes some of the technical complexity of working with Datalat away a little bit. And it helps you set up data sets that use Datalat, and then it's especially focused on if you want to run big processing on an HPC. And you can imagine you have all these histories and branches, and then bringing this back together if you've ever done complex Git merges and so on. That's not trivial, let's say. And so Babs is helping with this, and that's a good thing. Okay, so but how do you do the other side of it, right? So how do you say, this is how I did my processing, and here's how you can do it again. So the first thing you could do, again, Indie, I think, I like this a lot, but I want to also use it as a kind of a history in a sense. Did scripts, right? So there's scripts. If you go into the FCON 1000 project, or how did they do this, there's scripts. Scripts are great, but the tricky thing with scripts is if you want to run a second pipeline or a third pipeline, you usually have to write a new or different script, and there's not a I guess standard of how you run these things, and it can make it quite hard to scale up. If you're deciding tomorrow we want to run something different, we have run FreeSurfer, tomorrow we want to run QSI prep, you're writing a new script, that's slow, that's not so scalable. So lucky for us, again, our community, there's this tool called Boutiques, which essentially creates a language, a standardized language around how do you run tools. So it captures the interface, essentially all the parameters that you can tell a tool, and puts it in a standardized format. And what this allows me to do is call a bunch of different tools in a more standardized way. The other nice thing about it is that the developers can write these boutique descriptors for their own tools and then put them in a big community. So there's one on Zenodo, for example. And you can reuse these descriptors then. So if somebody does this for fMRI prep, then you can use these descriptors and say, how do I call fMRI prep very easily? So it takes care of the part, how do I call or make this tool run standardized? It doesn't tell you what options did I pick, right? So that's the last part. So this is where I come to a tool called NiPoppy, which is being developed now in a community that I think a lot of people are in our lab. And it really kind of does all of these things. So it takes boutiques, it takes containers, and it keeps track of when you're processing a thing, what parameters did you use, what software did you use with the container, and what version did you use. And I'm not going to go too much into NiPoppy for this. You should go into the afternoon talk when you see a full explanation of it. But NiPoppy really covers this whole scope in a way that I was showing you, right? Like, there's slightly different names here, but NiPoppy is a way that helps you to go from your raw data to bits, because it's helping you run all of these nice bits conversion tools that I was showing you before when switching between them, easy bits, Bitscoin, and sorry, not easy bits, Bitscoin, and then UDConf and all of these. And then for the processing part that I want to focus on here, what NiPoppy does is it essentially takes these catalogs of pipelines, and it makes it very easy for you to say, okay, I've set up NiPoppy for one pipeline, so what about a second pipeline, you know? And it has a pipeline catalog that allows me to take a description of the parameters, the pipeline, the software, and the version that somebody else has created, and pull that and run it on my data. And I can do this because my data are organized in a standardized way, and NiPoppy is aware of where they are and what exists there. Not sure if that's working for you. Well, guess not. Well, what you would see here if this was interactive is that there is a command that you can run that lists all of the pipelines that NiPoppy is aware of in a big Zenodo community. And then you can say pipeline install, and it'll fetch these three things, right? Software container with version and then the parameters. And then you can say NiPoppy run this pipeline, and it'll run the same way that it was run by the person who created it. So that's the answer to how do I make all my friends in the different places run analysis, processing on their data the way that I just did. And again, a lot of the motivation in our group is how do we work together in this open science way if the data themselves are not accessible, right? Because maybe they're in a hospital. Final thing I want to quickly say that NiPoppy does, which is very useful, is if you've worked with pipelines, even if they're comparable pipelines for processing, the output structure is usually quite different, right? So BITS has done tremendous work to figure out how do we standardize the input data or the not processed data yet, but then it's usually up to the pipeline developers what the outputs look like, and it's arguably not very realistic that they will ever agree on making this all the same, right? But what you still want to know is, has my pipeline succeeded? And so one of the ways of asking, has it succeeded, is you could say, did it crash? But another way of asking is, did it make the outputs that are important for me to continue with my analysis? So what you can do is describe for each pipeline, and by you, I generally also mean the pipeline developer or somebody writing a standardized processing I can say, okay, when fMRI prep is done for this subject, I expect the following files to exist. And that's essentially giving me the capability of tracking for which subjects and which sessions and all of this the processing has succeeded. So we're calling these automatic output trackers. And so I can run a command, for example, NIPOP track processing, and it'll just go into all of these different pipelines I've now run because my nice pipeline catalog exists, and it'll tell me for this subject and this session there was a success, or this is incomplete, or this is a complete failure. And then there's a nice dashboard that you can use and look at it and scroll around and says which pipeline succeeded in session 1 but failed in session 2 and all of these good things, right? But really what I'm trying to say here is when you standardize these things, that's when you can build tooling that really makes your life a lot easier. And so that's the real takeaway. And NIPOP is a nice tool that kind of holds your hand, or framework, I should say, through this whole process. Okay, great. So now we have tabular data, we have imaging, we have derivatives, and everybody is running things the same way because we know about containers and how to describe the parameters we're using to run them, and everything is golden. And now you might be asking, okay, well, how can people find my data? And so that's actually, you may have noticed, a thing that involves something we haven't talked about yet. So we have the derivatives in a somewhat standardized way, I would say, or as standardized as we're going to get them. We have the tracking, for at least. We have the imaging data in a standardized way because we're now all BIDS. But what about the tabular data? I haven't really talked about that. So BIDS tells you where to put tabular data, but it doesn't really tell you much about what to do with the tabular data or how to structure it, because everybody does it differently. So what I'm trying to tell you is you should harmonize your tabular data, and I'm going to try to tell you what I mean by this in the next couple slides. So the first problem is if somebody asks you for your data set, oh, sorry, maybe the real motivation, I mean, I'm sure you're aware of this, but if I'm saying find data, the thing that makes data interesting are usually the people that we took the images from, right? So if I give you an amazing T1 scan and you have no idea who it's about, it's not useful unless you're like pure methods developer. So I want to know about the people, and that's the link here to finding. Okay, so if your data set looks a little bit messy, you know, don't worry, that's very common. But that would be quite hard, both for a human and a machine to understand, right? So let's say somewhere in the middle of the study you have a new technical assistant started encoding ages, strings, or something like that. That's not great. So the thing is, you want to clean this up, and again, lucky for us, there are great tools for this. So OpenRefine is a very good tool that allows you to do things like little spelling mistakes or aligning things in columns into a standardized system. I'm not going to go into detail, but I would encourage you to check this out. This is a great software for that. Now, let's say you have done that, and now everything that is the same in your table is written exactly the same way, and it's all beautiful, and then your colleague says, hey, I'm trying to look at your data, and then it's supposed to be a human. Anyway, I don't know how that happened. So imagine there's another researcher on the other side. Then you say, oh, well, this is all you need. You just need my Excel spreadsheet, right? And then your colleague comes back and says, what does patient mean? Like, I don't know what you mean by patient. Like, what kind of patient, right? And then you realize, oh, well, you never explained to them what the data set is about. So then you get to the idea of data dictionaries. And data dictionaries are very, very powerful things. You should all make them. Again, our friends who have spearheaded all this have made data dictionaries. Now, the tricky thing with this data dictionary is it's a PDF, right? And so every study that makes a PDF data dictionary is a little bit different. So as a human, you can look at this and say, oh, that's great. I understand what this data set is about. But as BIDS teaches us, we also have to think of the machines. And the machines will have a harder time of parsing this type of data dictionary. So that's what I was saying about BIDS in the beginning. BIDS has these sidecar files, right? It also tells you a little bit how to structure them. So they're supposed to be human and machine readable. And you remembered this, and you say, okay, I will make my data dictionary a JSON file, and that's a good thing. So now you're writing, if you encounter the value of patient in my column called group, then really what this is a person with bipolar disorder or borderline, I don't know. Yeah. And a human understands this now, and that's great. You get a prize because you made a data dictionary. That's very good. But the interesting thing is, you might think, I'm done now, right? I'm ready. I'm happy to share with the world. And sorry, yeah, so now you're done, you're happy to share with the world, and you say, okay, please, I want to share this openly. And again, in our community, we have great, not tools, but platforms for this. So there are many different platforms that will take your data and then make them accessible to other people. They will do also other nice things, like take some of the data descriptions, you give them either on upload or through bids, and they will index it so search engines can find it. It's a thing called Google Dataset Search and such tools. And you know, that gets people to find your whole data set essentially, and that's very good. But what if you want to search for something very specific across data sets, right? So let's say you're interested in this very specific cohort. Maybe you're looking for people with a specific diagnosis. And ideally, if you search across many data sets, you don't want to do this by hand, because even if the data sets are described with data dictionaries, the tables still look different, right? So ideally, you want to ask a machine. And so the tricky thing here is if I encode, let's say, diagnosis and numbers, and you do it with these strings, the machine is going to have a hard time even with a data dictionary for humans. And so what you rather do is map to some kind of standard. You're kind of seeing the pattern here, right? Standards are good for solving these problems. So the ICD would be a good standard because these have numeric codes and definitions, and we can generally agree on what they are. So that's where we come to the NeuroBagel tool ecosystem. I'm not going to tell you all about it, but essentially NeuroBagel's main mission is how do we connect across data sets and make them discoverable even if they're not accessible. So I work at the Montreal Neurological Institute. That's a hospital. We cannot generally put our data on OpenNeuro, for example, but we would still like you to find the data. And the way NeuroBagel does this is use a lot of existing standards and then help people harmonize their tables or annotate their tables with these standards. What does this look like in practice? If you imagine that I'm in one institute and you're in a different institute, then, for example, this problem of diagnosis, we would say, can you please go to your big TSV file or table and find the column that has information about diagnosis? And you can do this because you know your data set, but I couldn't do this because I don't know your data set. So you do this locally. And then I ask you, oh, well, I see these values in your table, but could you maybe map them into an existing standard for diagnosis? So in this case, it's not the ICD here, this is SNOMED, but what you have to imagine is this is a standard that people have created over 35 years of work. So this is a huge body of these diagnoses. But what this allows me to do now, the machine understands that we both mean the same thing, even if it's written differently in the table, because it's a numeric identifier. And even if I make a spelling mistake and I say Parkins or something, and you do write it differently, if we both use the same numeric identifier, machine understands same subjects, same diagnosis. And we have nice graphical tools for this that help you do this. You can try those out. One slightly technical thing, I just wanted to show you how this actually works. So if you're familiar with BITS data dictionaries, you would be familiar with the gray part here. So you would say, if you encounter a column called sex, and you encounter in this column a value called F, then that means female, right? And so what we do after you have annotated your data with these standardized vocabularies is we write the same information for a machine, right? So here you say, you're linking to a very specific term that essentially is sex, and then if you have female, then there's another specific term from the National Cancer Institute, Thesaurus. I think they made this definition for female sex. All right, so that now means we have also understood the tabular data, so I can, when I'm searching things, I can ask, what phenotypic data do you have in this data set? What imaging data do you have in this data set through BIDS? What derivative data do you have in this data set? And I can ask this even if I don't have access to your data, because you have all these standards, and you can extract the metadata from these standards about what you have available. And that's the last thing I want to show you. So when you do this across data sets, you can make these kind of searches. So here I'm searching across a public portal that looks at different data sets from different institutes, and then I can see what pipelines have been processed and how many subjects have been processed with this pipeline, right? But I don't have access to any of this, right? These are actually clinical data sets. If I wanted to download them, I would have to go through a whole data access committee process. But because the standards exist at these locations, I'm now able to find a cohort that matches my research question before having access. And this is all through the power of standards. So my mouse cursor is stuck somewhere. Yes, all right. So yeah, that's essentially what I want to tell you. Through the power of standards, you get the power of metadata, which allows you to work in these spaces where we normally don't think of open science, right? If we think of open science, we usually mean open access, but I just want to say very clearly, that doesn't mean open access, right? You can work on these principles. In fact, you should, especially when you cannot share data openly. So final list of takeaways here. Make sure you use the standards that exist in your community. For us, BIDS for imaging, and then other standards exist. Capture your provenance, describe it in a re-executable way. Ideally use tools that help you to do this, because we don't have to reinvent the wheel, and this makes our research life a lot easier if we use these existing tools and ecosystems like NiPoppy. You know, there's a whole community. You don't have to describe your own pipeline. Probably maybe somebody else already did that. And annotate your tabular data as well with standard tools. And eventually, even if you're sharing this not in an open way, this makes your data findable, and that makes everybody happy. And that's how you all sit around the fir tree. Thank you very much.